Welcome back, friends. In the previous video about the DNA transcription, we have talked about the overview of the DNA transcription as well as some important properties. Now in this video, we will be talking about the initiation of DNA transcription, because we have seen there are three different stages of DNA transcription and we actually divided according to our convenience, like initiation, elongation and termination. So in this video, we will be talking about initiation. Now, to understand the initiation of DNA transcription, we need to know certain important facts again. These facts are, for the transcription start or initiation, they definitely require certain signal embedded in the DNA with which they know that this is the time to synthesize the RNA, right? So, some signals are required. So, once the signal is required, after that also how the process started. Like any other biological processes like DNA replication, transcription or translation, the initiation phase is the most important phase, because in most of the cases this initiation phase is itself a rate determining stage of all these enzymatic reactions, because these are also enzymatic reactions, right? Because enzymes are there, for example RNA polymerase in DNA transcription, right? So enzymatic reaction, the first stage is the most important. Again, in this case the initiation is very important, right? Because for the other stages like elongation and termination, they require too much of energy and all this process is going on inside the cell. So cell cannot back up once or back down once they start this whole process. So that's why initiation is done based on every measurement inside the cell, that this is the right time to synthesize. So the thing is, if I draw a DNA here, let's say this is the DNA, for the initiation part they require the signal for the start, and the signal is present in the DNA strands. Okay, now the protein in this case is RNA polymerase 2, or polymerase, simply termed as RNA polymerase. And the important property about RNA polymerase is RNA polymerase can initiate adding nucleotide sequence on its own, which is not possible if we use DNA polymerase in DNA replication, because DNA polymerase require a primer for the start, but RNA polymerase can synthesize RNA strand from scratch. So here, this is the DNA strand. The sequence that we are talking about is present somewhere, a particular region, and that sequence is termed as promoter. It is termed as promoter region of the DNA. Now this promoter region actually promotes RNA polymerase to attach and start the transcription of the DNA, right? And this promoter site contains a consensus sequence which is found to be conserved from many years. Now that conserved sequence here is present in two different actually locations. One is the location called -35, another one is called -10. Now what are those things? These things are the number of nucleotide sequences, right? And right after the promoter there is a sequence site which is called the start site. Let's say this is the start. The start site is known as a site 0, or say site 1, just right after that. So site 0 or say site 1, whatever you say here, in this particular case, this is the point of start. So in the sequence of this DNA it was written, then this is the site from where the transcription must start, right? So if we take that as a point 0, whatever nucleotide sequence present before that are termed as upstream elements. Whatever nucleotide sequence present after that are known as downstream elements. So whatever it is here are called upstream, whatever in this direction called downstream. These are the two different type of things. Now the upstream nucleotide sequences are termed as in the form of minus in prefix. In downstream they are termed as plus, or simply not written anything, right? So -10 means 10 nucleotide upstream of the start site of the transcription, right? Similarly, if I say 10 here or +10 here, that means 10 nucleotide downstream of the start or initiation site of the transcription. That's it. So the promoter region, as you can see, is present upstream of the initiation or start site of the transcription, and it is present between the 35 nucleotide to 10 nucleotide sequence upstream of the start or initiation site of transcription, right? And this DNA will go on, and ultimately after a certain time there is a termination sequence signal also. There is a consensus sequence found there also, right? But that's not a big deal now because we are talking about initiation only here. So once you know this structural feature of the DNA, now you need to know about the RNA polymerase enzyme. So the RNA polymerase enzyme, I'm not going to talk about the structures and everything because it's also having the structure of a palm and a finger, just like a hand, like, you know, palm, fingers and thumb. So structures like this, right? So different regions of this are destined for different tasks, right? There are particular sections of the RNA polymerase which helps in to melt the DNA. Melting the DNA means to separate the DNA strands from each other. There are segments of the, like the palm, which is present there, acts as a catalytic part, the actual catalytic part for RNA polymerase. So if I just draw it this way, so this is the catalytic part and these are the two other sections, right? Catalytic part means which is the part which actually takes place in the addition of nucleotide sequences there, right? So RNA polymerase singly, once it is a unit of enzyme, now this is termed as a core enzyme for the DNA transcription, right? And this core enzyme called RNA polymerase can bind with any DNA sequence. It can bind with any DNA sequence, no problem, just bind with it. But the problem with this core enzyme is that this core enzyme cannot recognize the promoter sequence on its own. It can bind, but it cannot recognize the promoter sequence. And if it is unable to recognize the promoter sequence, there is no way it can start the transcription. Okay. So for the recognition of the promoter sequence properly and also to bind with the DNA sequence tightly, it requires a factor, a subunit. That factor is termed as sigma factor. So sigma factor is a protein factor that is associated, that will be associated with the core enzyme of RNA polymerase, and which will help this RNA polymerase in two ways. One is to detect the promoter properly, and second is to adhere or attach with the DNA strand more firmly. So these are the two important tasks of sigma factor, and sigma factor is the most important factor in the initiation process because RNA polymerase is a common thing in all these stages, right? So what will happen here? So now let's draw it again. Now this is our DNA strand. Let's say this is the stretch of our promoter, and let's say this is the start site that we have already talked about, right? Whatever. So what happens now? RNA polymerase comes in, and RNA polymerase can actually bind literally everywhere. So RNA polymerase actually bind with it now. Let's say this is the RNA polymerase. It's actually majorly what happens. RNA polymerase not bind here actually. They bind with, let's say, RNA polymerase is a huge structure actually. It can cover a lot of nucleotide sequence, remember. So RNA polymerase start binding here in a particular area. Let's say this is the area. RNA polymerase binds here, but the binding is not firm. It is not detecting the promoter very well. So now sigma factor will come, and the sigma factor will help this polymerase to be attached and to find the proper promoter segment, right? So once it is attached, it is helping them to be adhered there firmly. So once it is done, now the next stage, what the next stage is very important, because in this case this polymerase kinds of change its structure, modify its structure a little bit, because upon binding to the promoter segment, what it does actually, it stretches itself when it is bound with sigma factor. Remember, when it is bound with sigma factor and detects the right promoter sequence, it stretches itself, right? So the stretching is very important during this region. So what it does, this is again and again, let's say this is the promoter and this is the start site. So the polymerase kind of stretches itself and sigma factor is still attached. So it will look something like this. Let's say, remember it was previously smaller, now it's stretching itself. Now in this condition it covers kind of at the very beginning when binding of it kind of stretches itself. Why it stretches itself actually? Because normally what happens, this polymerase along with sigma factor, they adhere somewhere very much upstream of the start site, even upstream of the promoter sequence itself, right? So upstream of the promoter. So to find the promoter you need to crawl a little bit, but remember when the sigma factor is adhered or attached with RNA polymerase cannot start transcription. Sigma factor is required to designate and determine the consensus sequence of promoter, but when the RNA polymerase is bound with sigma factor, they cannot start or initiate transcription or cannot move along the DNA. So during this part, the only way to detect where the promoter is, obviously the promoter will be downstream there, so they need to stretch itself. So now as you can see here, it stretches itself and it finds the complete promoter sequence from -35 to -10 and every region between that. And during this stretch, they can actually cover up to 60 to 80 base pairs actually. So you can see how much bigger RNA polymerase sequence can be, RNA polymerase enzyme can be, right? Now the consensus sequence that I've talked here, I didn't tell you the exact sequence pattern. The consensus sequence of the promoter, usually in E. coli it's found two different conserved units. In the -35 the units are TTGACA and at -10 the units are TATAAT. Now this -10 units TATAAT, it is also called as Tata box because you can see TATA, so it's a Tata, so it's called as Tata box. In the -35 that's called TT-GACA or simply TTGACA. So these are the consensus sequence. Now remember, when I say consensus sequence, it does not necessarily mean that every organism must have this sequence. It means this is conserved, so most of the cases we are going to find this sequence. It might have the TT, CA, C, something like that. It might happen like that, one or two difference in nucleotide sequence, but majorly it's kind of conserved. Now let's back here. Now what happens here, it stretches itself to form and find all this sequence TTGACA, TTGACA as well as Tata box. It actually covers every sequence here, 60 to 80 base pair sequence covering. So it will kind of eventually reach the start site here and finds the promoter properly. And once it found the promoter properly, there is the internal signaling process going on that this is the promoter and this is the start site, and now we need to synthesize our transcript or our RNA. So in that case, it needs to dissociate the sigma factor before it starts transcription, because if the sigma factor is adhered, it will not be able to transcribe, right? And how the signaling works and why sigma factor released from here, still completely not known. This is still a puzzle here, but the sigma factor will be released, and upon release of this sigma factor from here, let's say, the structure of RNA polymerase shifts back to the normal original form. So again, this was the promoter and the start site. So RNA polymerase now again shifts back to this regular structure of its, shrink itself again, and sigma factor released outside, right? So now RNA polymerase in a shape, a particular place inside this region, in the start site of this replication, the start site of this DNA. So remember, once it is in the start site of the DNA, upon binding of this, I mean the only way it can start, it can put into the start site is release of the sigma factor. But during this process, when it is stretched itself, it finds the promoter region, it finds the start site. During this particular stage, a lot of things are going on. Like from this actual particular place, they start to have a firm complex. Remember, I've told you sigma factor helps it to bind with very tightly with the DNA. So again, now polymerase 2 start doing some catalytic activities, like start melting the DNA. Now at the very beginning here, at this particular place, they form a complex where RNA polymerase is attached to the DNA promoter as well as sigma factor. That particular complex is known as closed complex or closed binary complex, right? After that, once the sigma factor is kind of losing itself and again it's stretching in, right? During that time, they start to melt this DNA a little bit, right? So let me better draw that as a melting condition here. So it is kind of melting the DNA. Start melting the DNA here, okay? And this is the polymerase. And sigma still, remember, sigma still attached there because sigma will be released just right after this. So DNA melting start to occur here, and if I draw it here, this was the promoter site, and these are the site for the transcription start. So melting is beginning here, and this is the stage. It is called open binary complex. So once the open binary complex is formed here, then what will happen? Once the open binary complex form, then this sigma factor will release, right? The sigma factor will definitely be released, and the release of sigma factor, remember I've given you the idea here, but now let's say it's releasing here because I've drawn an extra stage. So sigma factor is now released. Once the sigma factor is released, then what happens? It forms a lot bigger structure now. This is the polymerase now, right? So polymerase is in the right place here. Sigma factor is released. So now polymerase can initiate the transcription, right? And this complex is called as ternary complex. So remember, RNA polymerase is attached, then sigma factor helps it to bind with it, and it helps it to find the promoter. Once the promoter is received, I mean achieved, for achieving the promoter it needs to stretch the RNA polymerase to get there. Once it is there and find the promoter sequence and start sequence somewhere, provide some signal, and during that time they form the closed complex, tight complex. After that, they start melting, the RNA polymerase start separating the strands of DNA, and called the open binary complex formation is done. After the open binary complex is formed, sigma factor is released outside by a type of signaling. What kind of signaling? Still unknown. But after the sigma factor released, then what will happen? The polymer is in right place to initiate the DNA transcription, because it forms the open complex and they have all this this template DNA or template nucleotide there, right? Template nucleotide sequence there, right? So the polymer is having the template nucleotide sequence, and they can start adding the nucleotide sequence one after another to start synthesizing the RNA now, right? This is the position where they can start the elongation process. So till now every process here was the initiation stage, but still the initiation is not over. Why? Because at this place RNA polymerase is placed and DNA, it forms all this open complex, melting the DNA, it's done, fine. But this RNA polymerase is not sure of exactly what is going to happen. And that's kind of weird again, because during this time new nucleotide sequence will be given and it will start add nucleotide sequences, let's say 5-6 nucleotide sequences all together, and again this RNA falls off from this particular structure. It falls off from the DNA and again reassociate, rebind to the start site, again start transcribing, again releases the DNA, start transcribing, releases the DNA, come back and start again. So let's say it starts transcribing say 4 nucleotide stretch RNA and again falls, again come back and attach again, start again, falls. So they'll start at the very beginning, this RNA polymerase start producing small stretch of RNAs, for some times, for say 3, 4, 5, 6 times they are doing this. And during this time they produces a small stretch of RNAs which are not very much functional there as a protein coding unit, right, because they are terminated, and this situation is called as the abortive initiation of transcription. Now why abortive initiation is there, it's not still very much clearly understood, but the idea was that this small stretch of RNA sequences act as very different functionality inside the cell. Single stranded RNA can function differently inside the cell, right? But this abortive initiation is normally observed, and once the abortive initiation is overcome, I mean after 2, 3, 4 times, then the RNA is fine, the RNA can go for a long stretch and can synthesize continuously up to the sequence of termination. Okay, so this is how the initiation of transcription happens in cell. This is how it occurs inside the cell, right? It requires certain signals as a promoter, -35 plus 10, -35 to -10, and then start synthesizing it. Once it and sigma factor helps this RNA polymerase to attach and find the promoter. Once it found the promoter, it can actually bind with promoter more firmly and tightly, right? After the attachment with the promoter tight, the sigma factor will release and the RNA change in structure, again come back to the normal form, and then RNA polymerase melts the DNA sequence and then start transcribing the RNA in small stretch, abortive initiation, then again they will start synthesizing the RNA in full form, right? So that's how the initiation of transcription occurs in prokaryotes, and I hope that's helpful. Thank you. In the next video, we'll be talking about the elongation process of the transcription, so stay tuned.